Hello everyone, I'm Dr. Rehan Folari and it's my pleasure to welcome you back. And this time around, our discussion is going to center on the genetic control of cell growth, focusing specifically on these two pivotal processes, induction and differentiation, because these mechanisms are at the heart of cellular development and they are at the heart of the complexities that we know around different organisms. So let's dive deeper into how these processes orchestrate the behavior. So to set the stage, let's begin with an overview of genetic control, which is this intricate system that governs the regulation of cellular processes in order to ensure proper cell function and growth. So this system literally ensures that cellular processes occur in a well-coordinated manner and regulated manner. And within this framework, induction and differentiation stand out as very essential processes. Induction on one hand facilitates the intercellular communication in order to influence the fate of a cell, while differentiation allows cells to eventually specialize perhaps into blood cell, or whether the cell will become a neural cell or become a muscle cell, cardiac cell, epithelial cell, fat cells, or chondrocytes. So together, these processes drive the formation of tissues, organs, and ultimately help to form a functional organism. And at the heart of this, for each cell, you have a nucleus, which is a major organelle, right? And within the nucleus, you have chromosomes. And each chromosome, when you unwind it, you get to understand that it's actually composed of a tightly bound DNA double helix strand, and respective segment of each DNA strand is what constitutes what we know as the gene. So cellular growth is governed by the cell cycle, which is a sequence of well-coordinated and well-regulated phases. So during the G1 phase, which stands for gap 1, you have cells grow and prepare for synthesis of DNA. Okay, and during the S phase, you the DNA is replicated. That S phase stands for synthesis. Okay, so DNA is replicated. And then at the G2 phase, meaning gap 2, this ensures the cell is ready for division, which occurs during the mitosis phase. And the mitosis phase is what you're familiar with in your IPMAT. So checkpoints are also located at transitions between these phases in order to have them play a vital role in maintaining cellular integrity. These checkpoints help to ensure that DNA replication, is error-free and that any damage is repaired before the cell proceeds into division, thereby preventing genomic, instability and associated diseases. So generally, genetic control mechanisms are critical here in order to ensure that errors are corrected and that divisions proceed accurately. This regulation really prevents abnormalities that could lead to diseases like cancer, amongst others. So the basis of it's genetic regulation is actually fundamental to the function of the cell. So, and the cell does this through, control of gene expression. Cells determine which genes are active at any given time. And this involves what we know as transcription, where the DNA is transcribed into messenger RNA, and then translation, where RNA guides protein synthesis. So these proteins then execute, cellular functions, and this regulates it helps to ensure that cells respond appropriately to their environment and to the different developmental cues. So induction is actually a fascinating process where cells respond to external signals, thereby guiding their fate. And this mechanism is quite vital during embryonic development and tissue repair. For instance, salamanders, which is a reptile, can generate an entire limb through induced cell growth, a process that involves specific checkpoints and signaling mechanisms to coordinate tissue reconstruction. Here is an intact limb before it gets severed, And after the amputation, this is the stump you can see here, then the wound starts to heal, and this goes into this next stage where you have the formation of the blastema, and then the blastema starts proliferating, and you then start having patterning of the new limb tissue, and eventually a new limb is regenerated. So this remarkable feat actually can take several weeks to months, depending on the complexity of the limb and the environmental factors. And similarly, for humans, skin cells also proliferate and repair damage after an injury happens. So here is an immediate response within 2 hours. Remember, the skin is made up of the epidermis, the dermis, and then you have the hypodermis. So all through here, depending on the level of the injury or the wound, so you typically expect an immediate response. Immune response starts falling in place within 0 to 2 hours. And within the first 72 hours, the inflammation process is occurring. And within 3 to 10 days, you have proliferation of the tissues that will eventually replace the wounded area. And lastly, you have the retraction and remodeling occurring between 14 days to months, depending on the extent of the injury. So in humans, these examples, as well as that of the salamander, actually illustrate the importance of induction in maintaining and restoring. So the mechanism of induction in cellular processes are driven by a variety of signaling pathways and growth factors, all of which work together, to regulate cell behavior. And among the most influential of these pathways are the Wnt and Notch signaling pathways, both of which play very critical roles in determining the fate of a cell during development and in maintaining cellular homeostasis. I have an illustration here for the Wnt pathway. The Wnt signaling pathway, it's very essential, for regulation of cell proliferation, differentiation, and migration, particularly during the embryonic development. This pathway is activated when the Wnt proteins bind to a receptor complex that is composed of frizzled proteins and LRP5 or 6 co-receptor on the surface of the target cell. So this interaction, once it occurs, it triggers a cascade of intracellular signaling events. So in the canonical pathway, which is on the left side here, one of the key components is the B-catenin. Okay, and the B-catenin really interacts with transcription factors to initiate the expression of genes that are involved in cell fate determination and in stem cell maintenance. And at the end of the day, the Wnt signaling gets tightly regulated, and then dysregulation of this pathway would end up leading to diseases like cancer and developmental disorders. Similarly, in the in the Notch pathway, which I didn't put a picture of it here. It's another critical pathway similar to the Wnt signaling pathway, which regulates cell fate decisions, particularly in processes like neurogenesis, hematopoiesis, in vascular development. So the Notch pathway is activated by direct cell-to-cell contact between a cell and another, where a Notch ligand on one cell binds to the Notch receptor on an adjacent cell. And this binding is what triggers a cleavage event that would release intracellular domain of the Notch receptor, known as the Notch ICD, which then translocates to the nucleus. And in the nucleus, the Notch ICD interacts with the transcriptional co-activators to regulate the expression of genes that control differentiation. So the Notch signaling pathway plays a pivotal role in maintaining the balance between cell differentiation and stem cell self-renewal. And the disruption in this pathway would, just like the complication that could arise in the case of the Wnt signaling pathway, disruption in the Notch pathway are implicated in various diseases, including cancer as well and neurodegenerative disorders. Beyond this, we also have growth factors, such as, there are quite a number of them, and growth factors generally, including the EGF, known as the epidermal growth factor, here illustrated. Okay, they are crucial messengers that facilitate the transmission of signals across cells. These factors, what they do is to bind to specific cellular receptors on the surface of the target cell. And once they do that, then they trigger cascades of intracellular signaling events that end up promoting the growth of the cell, the differentiation of the cell, they promote survival and as well as migration of the cell. So through the activation of receptor tyrosine kinases and subsequent signaling pathways like the MAP, ERK, amongst others, these factors, they end up, they fine-tune cellular processes to developmental cues and environmental stimuli. So there are quite a number of these cells. You also have PDGF, which is the platelet-derived growth factor. So we have as well the TGF beta, that's a transforming growth factor. The beta transforming growth factor, which is in its case, it's very multifunctional in the sense that it regulates various cellular processes, including cell growth, differentiation, and immune responses. So it plays a role also in tissue development and repair. You may be familiar with the FGF, that's a fibroblast growth factor, which is involved in a wide range of cellular processes, including angiogenesis that have to do with blood vessels. It's also involved in wound healing, in tissue development, and there are multiple types of FGFs with diverse functions. We also have the IGF, that's the insulin-like growth factor, which plays a key role. It plays a key role in cell growth, in promoting cell growth and in development. It is structurally similar to insulin and is involved in the regulation of metabolism and survival. So we also have the VEGF. Okay, so VEGF stands for vascular endothelial growth factor. It's crucial for angiogenesis. Remember, angio refers to blood vessels. And so this VEGF is crucial for the formation of new blood vessels. So it plays a central role in various physiological and pathological processes, including development and tumor growth. We also have the NGF, which is the nerve growth factor. Okay, so NGF is mainly important for the growth, maintenance, and survival of neurons, of nerve cells. So it plays a very key role in the development of the nervous system. We have HGF as well. So HGF, that's the hepatocyte, and I mean, yeah. So the hepatocyte growth factor, which is involved in cell motility and morphogenesis, plays a key role in tissue regeneration and repair, particularly in the liver, like the name suggests. So we have BMP, we have so many others. Okay, bone morphogenic proteins. In their case, they are a group of growth factors that play a key role in bone and cartilage development, and they are also involved in various other cellular processes. So there is also GMCS factor, and that's granulocyte macrophage colony stimulating factor. So together, all of these factors that we have mentioned, the Wnt and the Notch pathways in collaboration with these growth factors, they all create an intricate signaling network that regulates the cellular processes with a high degree of specificity. The pathways, in general, they all ensure that cells adopt the correct fate. They ensure that cells migrate to appropriate locations, and they ensure that the cells differentiate into the functional cell types in order to maintain the integrity and homeostasis of the tissue. So that leads us to discussing more in depth about differentiation, which is the process by which unspecialized cells become specialized. That's what differentiation means. So it's very fundamental for the development of tissues and organs in order to enable them perform specific functions. For instance, nerve cells, right? Nerve cells transmit signals, whereas when you look at muscle cells, they contract in order to generate movement. So without differentiation, the complex organization and functionality of multicellular organisms will not be possible. So cellular specialization is ensured by this differentiation, resulting in the formation of diverse cell types, each adapted to perform specific. So genes are at the core of differentiation. Homeobox genes, for instance, orchestrate the body's structural organization during development, while PAX genes are crucial for forming specific tissues and organs, such as the eyes and the pancreas, amongst others. So these genetic instructions they enable cells to acquire their unique identities and perform their respective functions. Furthermore, signaling molecules are vital. They also stand as very vital players in genetic control. You have growth factors like the VEGF, amongst the many others we mentioned, stimulating cell proliferation, while cytokines and hormones regulate immune responses and metabolic activities. So these molecules really ensure that cells grow, differentiate, and respond, to their environment appropriately, thereby maintaining the health of the organism. So in order to study genetic control, scientists often employ techniques like PCR, which stands for polymerase chain reaction, to amplify DNA. All right. They also use microarrays to analyze the gene expression. And there is also the CRISPR, which stands for um, clustered, regularly interspaced, short palindromic repeats. Okay. So this refers to a naturally occurring defense mechanism in bacteria where you have segments of viral DNA that are stored in the bacterial genome. And this helps to then recognize and cut foreign DNA during subsequent infections. So the most popular or widely known application of this CRISPR is actually the CRISPR-Cas9 system, which is the most, because it utilizes this mechanism to edit genes by using guide RNA to then target specific DNA sequences and then the Cas9 protein to then cut and modify the DNA at those locations. So this tool has really revolutionized genetic research and gene editing in various organisms. So generally, these advances have transformed our ability to probe molecular underpinnings of cell growth, induction, and differentiation, thereby driving progress in genetics and biotechnology. So here is a further illustration uh, on how the CRISPR works. So here you have the Cas9 protein, which forms a complex with the guide RNA in a cell. Okay, so this complex then attaches to a matching genomic DNA sequence that is adjacent to a spacer, which is here in yellow. Okay, so after this, you then have the Cas9 RNA complex cutting the double strands of DNA. And finally, you have the programmed DNA, which may then be inserted at the point of the cut. So that way it has really allowed for a lot of manipulations genetically. So from the foregoing, we will agree that genetic control has proven to be very integral into understanding diseases because mutation disrupting genetic regulation can result in cancer or inherited disorders. So that's why insight into genetic control remains indispensable for comprehending and treating a number of these abnormalities. Also, advances in gene therapy and personalized medicine have enabled targeted interventions, thereby offering hope for conditions once they are deemed untreatable. So the future of genetic control research is really exciting, particularly with the CRISPR leading the way in genetic engineering, because it allows us to make very precise modifications and thereby holding a lot of immense potential for treating a lot of genetic diseases. So if we are able to combine genetic research with nanotechnology as well, then this could yield a lot of groundbreaking diagnostic and therapeutic interventions thereby paving a lot of ways for new discovery. So while the potential of genetic manipulation is quite vast from all of what we have discussed, it's also very it doesn't go without saying that it possesses ethical challenges and questions around safety, consent, and long-term impacts must therefore be addressed on a number of these procedures. And it's very pertinent to also balance innovation, as we would see in genetic research, with ethical responsibility in order to ensure that progress benefits the society as a whole and that it's not skewed just along the scientific discovery and possibilities. So, in conclusion, we understand that genetic control is the cornerstone of cellular processes regulating growth, induction, and differentiation. And as research advances, we gain opportunities, to tackle diseases, enhance therapies, and unravel the complexities that we have in life generally. So, I thank you very much for joining me in today's exploration of genetic control in induction and differentiation. That's it for today, and I hope you found the lecture quite insightful. So, if there was a particular section that resonated with you, please let me know in the comment section. And I thereby encourage you to stay tuned for more in-depth discussion on developmental biology and on anatomy topics generally. I particularly would like that you feel free, to share your questions or comments in the comment section, and I'll gladly continue the conversation with you. So until next time, stay curious and keep learning. Bye.